I'm so thirsty. I didn't have any breakfast this morning.
I love milk. I have every morning.
Me too.
Do you know how your body breaks down milk?
No? How?
It looks like we have a new field trip today. To the bus! Hold on. It's gonna be a bumpy ride. Through the mouth, into the esophagus. We are now entering the stomach.
What is that?
Why, Nadine, that's a piece of pizza. But just ignore that. We are now entering the small intestine. Folks, look out the window. One enzyme we see here is a prokaryote called E. coli.
Go.
E. coli lives in your intestines, and what's fascinating is whatever you eat, it eats. When you drink milk, the E. coli will break down the lactose. This is a gene. This is where the lac operon is produced and regulated to break down that milk you just drank. In prokaryotes on the gene, you will find what's called a promoter, and an example of this is a Tata box.
Why is it called a Tata box?
Good question. It's called a Tata box because this is an area of the gene made up primarily of alternating adenine and thymine bases, which create the sequence Tata. Now, where was I? Oh, right, regulatory sequences and promoters. This promoter region is where the RNA polymerase latches onto the gene to express it. In bacteria, they have not one, not two, but 3 genes next to one another to help break down the lactose.
What's this thing?
That there, squeezed in between the promoter region and the genes, is a thing called an operator. An operator is like a light switch. It's either on or off, which means those genes are either expressed or aren't expressed. Now, another piece to the puzzle is a repressor, which fits right onto the operator. If it's there, RNA polymerase, it cannot latch onto our gene, and our genes are in the off position. However, once you drink a glass of milk, and the lactose appears in the body, and the lactose molecule fits perfectly into the repressor, and the repressor goes away.
So now that the repressor is gone, what can latch onto our gene?
Oh, I know. RNA polymerase.
Very good. I'm glad you were paying attention. Now that RNA polymerase can latch on, what can happen to our gene?
mRNA can be made, which means that a protein can be made during translation. This will break down the lactose from the milk I drank. Yes.
Way to go! Now, what I just went over is called positive control, or the genes are expressed only when an active regular protein is present. The opposite is called, give it a guess.
Negative control?
Yes! Can you guys guess what negative control is?
Is it when genes are expressed when there is no active regulator protein present?
Almost. Here's an example. The TRP operon is actually off in the E. coli when the TRP chemical is present. If TRP is present, why would the bacteria cell want to make more TRP when there's already some available? Would you want to buy more shampoo if you already have 2 full bottles at home?
No.
Exactly, except if you have no TRP in your system, what the bacteria does is change the shape of the repressor, the way the RNA polymerase is able to latch onto our gene and make our proteins. The proteins eventually make TRP molecules, and everything in our body can continue in its cycle.
No, but can we get out of here? I don't feel so good.
Of course. Hold on tight. Back to the esophagus we go!
Oh boy.
Now everyone should know that gene regulation is needed to produce important proteins. Hope you enjoyed our trip through the human body. Next week, we're traveling back into the body to examine the phases of meiosis.
Oh, great.